Well, hello and welcome to Lab on Got Protein. And yeah, that's Arnold. And for most of you, you're much too young to remember the commercial, but it's the ladies from the old Wendy's commercial. Where's the beef? And most of us, when we think about protein in our diet, this is what we think of. Well, actually, I think of what's at the bottom of the page, but we think about the eggs and the cheese and milk, and of course, the animal. And turns out our best source of protein is not the animal products. So let's go into the matrix and see. So there's a picture from the Wendy's commercial from the 70s, and the woman we're calling up, where's the beef? One of the best sources of protein is actually the picture behind, which are the legumes. That's one of the challenges that come up. And this is from Vegan Street, it shows at the bottom, but the amount of protein. So I don't eat meat usually, although today is a special day. And so we're going to have salmon. And so today is Joey's birthday that I'm recording this. And so I'm making his favorite meal, which does include salmon. But that shows vegetables actually are perfectly packaged by Mother Nature with protein and carbohydrates and water and a wee bit of fat. And so just an interesting, in case you've never heard this. So when people don't eat meat, everybody says to them, where are you getting the protein? It's in the vegetables. It was already there. This, in case you've never, most professional bodybuilders and a lot of professional athletes, and a lot of them don't want people to know that they're actually vegan because they find they have faster recovery, they're stronger, and they have longer endurance, and they don't want the other athletes to know their secret because they don't want them to do it too. They want to keep the advantage. And so there was an interview I heard from a guy who was a lineman for the Oakland Raiders, and the other teams could never keep up with him. By the third quarter, he was still going strong, and they interviewed him, and he finally, at the end of his career, told his secret, which was he was vegan. And he was strong. He was huge, like this guy. And yeah, nuts, legumes, and your vegetables, and they're Woody and Buzz Lightyear. So what are proteins made of? This is what our lab's about. So proteins are made of amino acids. So the amino group is the amines that we just learned about in our organic lab, the nitrogen compound, and the carboxyl group or carboxylic acid. This picture is shown by your teacher who is a biochemist, and this is what they actually look like. They are ions. Nitrogen attracts the hydrogen, and the carboxylic acid, the acid gives up the hydrogen. And so they're actually Zwitter ions is what they're called. The R group is variable. So sometimes it's a carbon, sometimes it's another carboxylic acid. So there's 20 different ones, and they hook up by a reaction that happens from the amino group of one to the carboxylic acid group of the other, and they form a polymer, which is a long repeating chain. So just like us holding hands, which, oh, maybe I'm a dreamer, but it's going to happen again. So this is a picture. So when a biochemist has trouble falling asleep, we just count our amino acids. I have no trouble falling asleep, so I don't have to go there. But the piece that's sticking down is the variable part. And you can see the ones at the top that have the green highlights, those all have carbon-hydrogen side groups, so they are non-polar. In the middle, the purplish lilac ones, you can see they all have an OH or an oxygen. They're going to be able to hydrogen bond. They're polar. The one with the sulfur in the middle is special. And then at the bottom, we call them electrically charged or acidic and basic. And so they have the negative or positive extra negative and positive on their side group. And that negative and positive is going to create an ionic attraction. And these attractions, the nonpolar attraction, which we actually call hydrophobic, they're not actually attracted to each other, because as we're going to learn, nonpolars are not attracted, but hydrophobic means they don't like water. And we're talking about in our bodies, and our bodies are water. And so that need to get away from water actually makes this one of the strongest forces that's going on in proteins. So these guys are all going to accumulate in the middle of the protein because they're getting away from the water. They're hiding. And so they all end up together. The H bonding, and then we're going to see the ionic. And so this is why this goes with the intermolecular forces. What you're going to be doing, we're going to go through these different forces, but what happens in proteins is proteins have these beautiful structures. So if I was a true biochemist, I would have artwork in my house of proteins. But we're going to walk through, or I'm going to walk through right now, these 4 different layers of structure. And no, you're not going to be tested on it, but this is on your lab. And this is hopefully a couple of you, I know several of you are interested in this, and some of you will be going to higher level classes in biochem. So the first level is called the primary structure. And this guy won a Nobel Prize in the 50s for determining the first protein structure that was determined was insulin. He then also determined the DNA sequence, because our DNA, 2% only of our DNA codes for proteins. Again, we're the only species where it's only 2%. The other 98% in humans is where the interesting part comes at, and that's quantum. We should be calling it quantum biochemistry. But anyway, he determined the sequence also of the gene, the DNA part that codes for this. And so we were able then to create human insulin protein for people who are truly diabetic. But the primary structure is the sequence of amino acid, the order they are in. These 51 amino acids, you can put them in any order. There's only one way in which it comes out as insulin. So there's something like 10 to the 81st power of different ways you could put these 51 amino acids together. This is a really short chain of amino acids, but that connection between the holding of hands of the glycine, leucine, serine, aspartic acid or aspartate, that is a covalent bond. That is a peptide bond. Covalent bonds, you're not going to break them in this lab. This is just showing you some of the different proteins that you may have heard of. And these guys get huge. So molecular weight is the AMUs or grams per mole. So insulin's a small one, and it's 6,000. I think of water, which is 18, and that's how many amino acids. So the largest one in our body is in our muscles. So myosin is one of the proteins in muscles, and it's just huge, 800,000 grams per mole. Imagine doing that math, right? The secondary structure, so the ribbon that you see in the upper right-hand corner, that is the primary structure. That's the covalent links, the peptide bonds that are hooking everything together. And then the dots are the hydrogen bonds between, this is actually the backbone has that oxygen double bond and the nitrogen with the hydrogen. And this guy, this is Linus Pauling, holding the orange because he is the vitamin C guy. And he is, we've talked about him several times in this first half of the term because it is really all about him. He had a cold, and he was in bed, and he said to his wife, hey, bring me over the structure stuff I was working on. And he sat there and cut it out. They didn't have, tablets and stuff. So he wrote it out on a piece of paper and then cut it out. And then he realized he had this repeating double bonded oxygen and nitrogen, hydrogen that came near each other. And he said, well, they must have an attraction, this hydrogen bond. And he came up with the idea that alpha helix, and that's what won him the Nobel Prize. It was the first helical structure. And then after that came DNA and all our other helixes, lots of helixes. So if you have curly hair, like I do, your curls are alpha helix. There's also the beta pleated sheet. And so what is happening is this determines how strong, how flexible, how soluble the protein is. So alpha helixes are much more, but it is hydrogen bonding. And so if you can jiggle and get things up to a high enough like heat, it's going to jiggle this apart. And so that's one of the things that happens when we cook is we jiggle apart the secondary structure because the heat is going to overcome that, but we cannot jiggle apart the ribbon. And so this is a picture of a prion. And so on the left of the picture, you see the beautiful alpha helixes. That is a normal prion. And then what happens as soon as one changes, every single prion, it's a mini protein, a mini peptide actually. They don't call it a protein. It reverts to a beta sheet, which is another form of secondary structure, and it causes damage. We'll see a picture of that in a moment, but first I mentioned, so Linus won the Nobel Prize in Chemistry in '54, and then he won the Peace Prize. And that is also, I love this picture because it shows his wife, and he gives full credit to his wife, was always there supporting him, nurturing him, and she was very active in the peace marches also back in the '50s, which they then took his, our government again rewarded him by taking away his passport. All right, another example, these are examples of alpha helix in our body is myosin, our muscles. So long fibrous things would make sense that they would be. Also, our blood vessels have a triple helix, which is collagen. And so our hair is keratin, alpha helix. So our hair can be styled in different way. Our muscles, that's me. I thought that was impossible, but I finally learned how to do that yoga pose. Beta sheet is another one. And so these little purple, let me see if I can get my laser. I am not good at getting the laser, but in the lower corner, the purple he's drawing in. So the black lines, sorry, let's go back for a second. The black lines, maybe not. There we go. The black lines are covalent bonds. We can't jiggle those apart. But the purple is the attraction, and that attraction is strong, not as strong as a covalent bond, but it is strong, and it's holding these different parts together. And we get this pattern that makes what's called the beta sheet, and it is found in silk. And so silk, they are actually have found type of silk, and it is actually stronger than Kevlar. It is a protein. It's lightweight. It's much lighter weight than Kevlar. It's flexible, and it provides a high degree of protection. So strand per strand, it weighs so much less, 3 times more flexible than Kevlar, and also is 5 times stronger than steel. And it is what they're trying to make. I don't know enough about this. If you don't have a topic yet, you could definitely look into this. Damon mentioned it in his innovation. So, you know, when we get to the innovation projects, you could pick one specific innovation that comes from nature, called bio innovations. We'll talk about that in another slideshow. But it naturally is capable of absorbing a huge amount of energy. And so farming spiders is not viable because spiders don't play nice. They actually fight with each other, especially the type. Every spider web is slightly different, every type of spider, and it's this specific spider really fights. But spider webs are freaking amazing. So scientists are trying to splice the gene into silkworms because silkworms actually play nicely, and they also have spliced it into goats. Weird, but then they milk the goat, and this I don't have a picture of it because it's really bizarre, but you can Google it. All right, so it's biodegradable, so it's our new silk. So if you think of fishing nets or fishing lines, we're not going to have that pollution that's happening. Airbags, parachutes, it's going to be made with this silk, and also sutures. So there you go, Captain America, and our new tendons and ligaments. So when we have to have surgery, so they can make it so it's not going to dissolve in your body, but it's natural, and so it's going to be healthier for our body. I mentioned prions, so silk could do that as your topic if you still don't have one, or even if you have one and you're inspired by this. If you want to do a downer topic, you can pick prions. This has got to be one of the most downer topic. This is mad cow disease, in case you don't know what it is. And again, the normal prion is the blue one. And then somehow, as soon as one is exposed to a diseased prion, which comes from animal, and it's in our brain, and it changes. And this is what ends up happening. So the blue, purplish blue is our normal brain tissue. And then that is when you start having the prions. You get these holes all over, and eventually there's too many holes. And the thing that's scary, so this one, I'd have to flip it, but then the words would be opposite. So on the right side this time is normal, and the left side is not. In older people, even older than me, because as you get older, like myself, you have more connections between neurons, and so the holes take longer to see the effect. And they have found like a significant percentage of people who die from Alzheimer's actually have this. The symptoms are identical. In young people, because your neurons are connected different, you die pretty fast because you go senile and die. And so that's the good news. You don't have to go this long, awful path. But the bad news is, right, your brain's destroyed. All right, tertiary structure is the overall folding. It is this beautiful folding, and this is back getting with the lab. And it's due to the interactions. Interactions mean attractions. So remember we talked about the different types. And so we see the alpha helix is an area of pattern, but it's the overall. And that shape actually determines the function. That actually has very specific function. There are fibrous ones, so the fibers like our muscles that I showed, or there's globular ones like our immune system. And so it folds. So there's the strand that is our primary structure, and then somehow it folds, and it only gets one go at folding. And then once it gets unfolded, it never figures it out again. All right, so this is what you're going to be asked in the lab, is what ones are disrupted. And so these are the 4 types. So we have disulfide bridges. That is covalent, only happens between sulfur groups. The only thing that breaks that are the heavy metals, mercury, silver, and lead. And this is the problem. There are a lot of sulfur bridges, disulfide bridges in our brain. And mercury fillings, the silver fillings are mercury. They just have a silver color. That is right by your brain. And it makes, when you chew, your spit bacteria creates methylmercury, and methylmercury can cross the blood-brain barrier, and it binds the sulfur, and you lose the bridge, and you lose the tertiary structure. Now, unless you have like colloidal silver at home, I think somebody had that one time and they tried this, you might be able to denature. I'm not sure how many disulfide bridges there are though in eggs, so you're not really going to get to mimic this in the lab. There are the ionic attractions, and so the salt bridge is what it's called. And any way we can change this, so a change in pH, this is how you're going to curdle the milk, is going to interrupt these, and it comes apart. The ribbon stays, the primary structure stays, but the secondary, tertiary structure is gone. On C, that's our hydrogen bond, and the hydrogen bond gets disrupted if we add something like an alcohol. So rubbing alcohol, that was the idea behind the hand sanitizers, which have been shown don't do what they think they do. What is better for viruses and bacteria and just keeping contamination down is to disrupt the hydrophobic. So the hydrophobic are inside in the middle of the protein. They're not really attracted to each other. Notice there's no dots on D in that lower right-hand corner, but they don't like the water, so they're found on the inside. So the whole inside of the protein is these. So if you use soap, soap is hydrophobic to it, and the soap pulls this apart. And as it pulls it apart, it thinks there's something safe on the outside, that the water's not there anymore. And so the hydrophobicity, so soap has both hydrophobic and hydrophilic. And so it pulls it apart and it all comes apart. Not the ribbon stays together. We'll go through that again. So this is the overall tertiary. And again, the middle parts would be the hydrophobics. And you get this beautiful structures that we can make artwork with. There's also a 4th level that happens in really big ones, because the really big ones, to make them as one is too big. And so our body will make them as 2, 3, 4 units and put them all together. And then there's attraction, usually hydrogen bond attraction. And so it's multiple polypeptide. So again, the primary is covalent. The secondary is hydrogen bond. The tertiary is hydrogen bond, salt bridge, hydrophobic. We'll see this again. And then the quaternary is when we just have multiple ones that are attracted to make the overall protein. All right, so there's two ways. This is what happens in digestion. Digestion within your stomach has what are called proteases, and it cuts. It doesn't cut one at a time. It cuts a couple off, and there's a couple proteins, and eventually get single amino acids. And so what happens though, this is what your proteins look like. They look like a ball of string, and this poor cat's going, I can't get it apart. So what also happens in your stomach, in case you don't know, is your stomach has a really low pH, lowest pH in our body. Lowest pH probably on the planet. It's like a pH of 2. It's extremely acidic. And what that acidity does is it denatures, it allows it to start unfolding, so unraveling there. And then the enzymes can get in and cut it apart. So in this lab, we're not going to use any enzymes. There is in making the hard cheeses like cheddar cheese and stuff, they do use enzymes as well as pH. We're going to actually, in this lab, denature. And so the normal protein is denatured and unravels. So like the kitty cat there, and renaturing. This is the thing that's something interesting. So not as easy to do, but typically once the egg is denatured, or the milk, and so you're going to have to sacrifice an egg. It's the egg white we want, because that is where the albumin or the protein is. The yolk, something different. And you're going to denature it in different ways. So one is you're going to simply agitate. So you just need, I think, 6 glasses. If you have shot glasses, that's great. Or you can get little Dixie cups. And you're going to split your egg white. You really only need 1 egg, but if you want to use 2, you don't want the yolk in there. Give the yolk to your dog. They love it. Makes their coat shiny. And you can agitate. So agitate is you're just jiggling and giggling it, and that's going to interrupt the H bonds. If you had a heavy metal and you want to make it go crazy, that's what the Mad Hatters went crazy, because the mercury they used to make the felt in the hat just right. Dentists also, in the past, all had shorter lifespans, and people working in pharmacies because of their exposure to the heavy metals. A lot of dental offices are mercury free now, and so that's use a different type of filling, the composites. Alcohol. And you can use rubbing alcohol, you can use vodka. Beer and wine are not high enough, but if you have hard liquor, you can actually use that. In the lab, you just tell me which type you used. Doesn't matter, tequila. You're not going to pee on it, but well, I suppose you could do a mystery one. But you're going to use either lemon or vinegar or some kind of acid. And so going back to the alcohol has OHs, so it's going to pull the hydrogen bonds. It's going to have an attraction to the H bonds. The lemon or vinegar is going to disrupt the ionic interaction, the salt bridge. And then you're going to use either a detergent or soap, and that's going to pull the hydrophobic part out. And then you're going to try and heat it up. You try in the microwave, you can fry your egg and then eat it. Doesn't matter to me. Everybody had fun doing it different ways. And of course, it denatures. You've all seen a fried egg or a scrambled and fried even more, because you're just using heat. You're heating it up. You can just boil it. So you can put if you have depends on what kind of glass you have or test tube. So that's one part. You're going to do that's the egg 6 different ways. And then you just take a picture of your 6 eggs and tell me which way worked best. The 6th one is actually a mystery substance. So some people found some really cool stuff. So it could be fingernail polish remover, it could be Drano. There's so many, people came up with really clever things that I would never have thought of. The other part of the lab, actually the first part, is you're going to make milk curds. And there is a bonus point. If then after you make your milk curds, take a picture of you making your milk curds, you can make it into some beautiful sculpture. You can try it, just a small taste, or you can have a lab partner, your dog, your children. Some of them will try it. This is me when we got to be in lab. There's a picture of the lab. And I can't do that amazing artwork. And I also didn't make these crazy puns up. You can always write a pun or a poem also with your lab. But yeah, and the fairies are there. But what's in your milk? Well, which of these are the protein? And it is number 4 and 5. So I guess those are the greens. Most of milk is water and then fat. Lactose is the sugar, but we're going to go after the proteins. So that's what this lab is about. So the proteins in milk. So little Miss Muffet sat on her tuffet eating her curds and whey. That's what we're making is the protein. We're isolating the protein. And we're going to do it by denaturing it, not hydrolysis. So that is the curds, which is the curds. If you ever been to Tillamook Cheese Factory, and some people will take it, is Casein. This is Joey 10 years ago, 11 years ago in Nepal. When we were in Nepal, we got to go to the cheese factory. Got destroyed in the earthquake 5 years ago, and I'm sure they've rebuilt it. And they all called Joey his brother, their little brother, and they absolutely loved him and adopted him. And that is a giant copper pot, and Joey's stirring the curds. Casein is also used in paints. It's also used in toothbrush bristles, and is also whipped cream. And there's a reason Elmer's glue has a picture of a cow. It is actually casein. It was traditionally, you can make glue. You can make this into glue. I think it's sodium bicarbonate, baking soda you add, and you make it into glue. There's actually, we did this last year before I got kicked out of being on campus, and we glued a bunch of Valentines. It was Valentine's Day, and so everybody made a Valentine, and they're still there on the wall, and so it's awesome. So if you want, you can make a Valentine, and I'll go and glue it in, or I can sneak you in. Oh, did I say that? All right, so if you want to make the glue, you can just Google it. You go through the recipe the same way, and then you add sodium bicarbonate. And then, of course, it's used to make cheese. So this is something, in case you didn't know, most people know about the lactose intolerant, but it's not actually the lactose you should be worried about. It is the casein. So there is a very dark side to cheese, very dark side. There is a reason people don't want to give it up. People are like, okay, I'll give up the dairy for 30 days, but not the cheese. People give up meat. People give up eggs before they give up the cheese. And it's a stepwise thing, but there's a reason. One, casein's really hard to digest. It has to do with, it doesn't denature very well in our stomach. And the enzymes can't cut it apart quite right. It creates excess mucus. So I learned this 25 years ago. First day of acupuncture college was when people have mucus, which is one of the things coronavirus makes, you have to stay away from dairy. Dairy creates a lot of mucus in our body, and so that phlegm that builds up. It has now been altered by how we ultra homogenize our food in the high heat. And right, so the spider's telling little Miss Muffet, it's not good stuff. And so the partial digestion ends up causing stomach issues. You all know people who go, oh, I can't eat that, I have stomach issues. They don't want to admit it's actually from this. And so you end up with constipation, gas, bloating, diarrhea. Lactose gets blamed for that for reasons, but casein's a bigger issue. It also causes a lot of skin issues. And for girls, you give up casein, you give up cheese, any kind of cramping you have with your menstrual cycle, it goes away within a month. It goes away. Yeah, but they don't want to tell you that. And the reason it's so hard to give up, it's an opioid. It's addictive. It's actually considered one of all the opioids are addictive. And so if you're somebody who's like, no, I'm not giving up cheese. Cheese is okay. Just like 3 times a week, you got to give it up. And there's going to be a 30-day extra credit challenge coming up. That could be it. I'm going to do it. No cheese. You can have pizza without cheese. I don't like the vegan cheeses, but pizza without cheese is amazing. And there's Bessie telling me, yeah, she's looking at me like, what are you telling them? Oh, but the whey protein doesn't get off. So any of you who are into bodybuilding, you probably are like this guy, he had the big bag. So after the milk is curdled, you're going to actually separate the curds, which is what you're going to make something out of. And there's the whey, you're going to drain that off. So there was all this whey that was wasted and they figured out a use for it. They said, hey, it's protein and it's in everything. If you eat processed food, there is milk isolated protein in there. That is the way. It's lactalbumin, which means milk albumin. Is it healthy? Oh, well, studies say that it will cause your muscles to get bigger, right? So it's high in those branched amino acids. There's a whole thing about this, blah, 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 blah. And then, oh, there's a little word about, by the way, yeah, much higher rates of cancer. We're not talking like 1%. So it was actually, there's a big study actually that's been going on for at least 10 years. I think it might be 20 years down at UCSF. I don't know why I can't think of the name. But anyway, this was after World War II when we went in, bombed Hiroshima. Then we went in and said, we're going to be your friends and we're going to introduce the American diet. And so Japanese men started drinking cow's milk and eating cheese, and they had a 20-fold increase, 25-fold increase in prostate cancer. And what happened, they realized, was their diet had changed dramatically. They had increased the amount of eggs and meat, and a 20-fold increase in, holy cow, go for it. So this was done in culture, in Petri dishes with prostate cancer cells, and they dripped organic cow's milk, and they only saw a 14-fold. Oh, sorry, they repeated it 14 times. They always saw a 30% increase in the cancer cell growth, and how fast it went to metastasis, even more scary. They tried it with almond milk and other milks, and they actually suppressed the growth. In a meta-analysis where they just look at all the data, they found, oh dear, this is actually happening everywhere. The anti-cancer foods are never dairy or eggs. There's no animal products there. There are sweet potatoes and broccoli, sulforaphane, really cool topic. Yeah, or any of the fruits. All right. So, remember that? That will be, you could do that. You could be like, I am going to eat 3 pieces of fruit a day. Comes up. I'll give you the extra credit if you want to try it. So how did little Miss Muffet make her curds? That's what you're going to do. Casein forms my cells, which are these large aggregate molecules. So there we go. That is what a curd looks like. That's the casein. And when we drop the pH, which you're going to do with either lemons or vinegar, you're going to try it 2 different ways. And I sure hope I'm recording. Don't want to do this again. Anyway, it denatures. And there you can see that's the picture of how they say it denatures, and you get curds, and then you separate them. So it denatures, and this is cottage cheese, sour cream. It's cheese curds. Cats absolutely love it. So if you don't want to eat it, your cat and dog will love it. Or you can make it into plastic dice. We've made it into lots of different things, and students have made really cool stuff. They used to actually, the first plastics were actually milk curds. The first buttons were actually, in the 40s, the first plastics before the plastics came out in the 50s and 60s from DuPont. Buttons, decorative buckles, beads, jewelry, fountain pens, the backing for handheld mirrors, and fancy comb and brush sets. Yeah, apparently the Queen of England actually had plastic milk jewelry. There you go. This is the guy in the basement. Can't go on the tour anymore, so you don't see him, but he's still there making the fancy jewelry. And that's me in the lab, making myself my pink earrings, right? Not really. We made GAC, so fun. You can make it from milk, starting from milk, and then you go to the glue, and then you add boric acid. And right, next term, those you're going to be in 223, you get to do a demo for the class, since it seems like we're not going to be on campus again. So the skinny and skim milk is they say they remove the fat, but I think they really remove the gnomes. They say the protein's the same, and actually since we've been doing this, it has worked. We've been able to denature it. The first year I did it 5-6 years ago, the skim milk, there was no protein in it. We couldn't get any protein out of it. But now, so they must have changed somehow how they were ultra homogenizing it. These are different things you can try to lower the pH. I will tell you, the Coca-Cola and Pepsi, you have to add a lot. We used to get it to work and we don't anymore. We couldn't get it to work. You can squeeze an orange on there or use orange juice. Actually, cranberry juice works really well, and Red Bull will do it really well. You have to add more than with the vinegar to lower the pH, but the Red Bull works really well. I would imagine coffee should work really well. You have to add more than normal and you curdle it and denature it. So if you want to keep listening, why you should give up the animal products for 30 days. This is the prostate study from Harvard. Men who ate 2.5 eggs per week had double the cancer progression, metastasizing to the bones, 81% increase in death. Only 2.5 times a week. What do you think was the only thing worse? One guess? Oh, you were going to say dairy. Turns out, no. Chick-fil-A has done a good job at making sure you don't know this. The only thing better than eggs at doing this was actually eating the chicken. If you want, barbecued chicken is the worst of them all. Chicken actually 4-fold increase in cancer progression. And it is because when you cook the chicken is really high in these amino acids, and it makes, when you heat it, you make nitrosamines, so the heterocyclic amines. So if you want to talk about nitrosamines, it's in poultry. And when you cook it with sugar, which is barbecue sauce, is pure sugar, you're guaranteed. It's a class 1 carcinogen with plutonium. Might as well add plutonium to your chicken. Get it over with. And you also make age. Eat. That's from my garden. That's my dog. I'm really short, like the size of a gnome. There you can see my countertops behind there. So I'm like 5'3". My dog's really small. She's under 10 pounds. And all right, high protein diets. Average American gets double what is actually recommended. And it acidifies your blood. You guys don't want acidic blood. It is the cause of acidic blood. It pulls calcium out of your bones, leads to arthritis, fractures, osteoporosis, and cancer. Cancer can only grow in acidic blood. We are talking 7.2. Normal blood should be 7.4, and you get that from eating fruits and vegetables. So acidic doesn't mean 2 here. It just means lower than normal, which should be 7.4. You also get higher exposure to toxins, antibodies, growth hormone, pesticides. You get degenerative diseases. Cannot recommend this book enough. Damon, you got to read this book. It is like all the books that keep coming out on nutrition and the importance of more fruits and vegetables. So that's T. Colin Campbell. I can go on. My other classes is all we talk about. So it was the biggest study that's ever been done. 17-fold increase in heart disease from animal-based protein. And then it destroys your kidneys, because your kidneys is how you get rid of the nitrogen. And it also makes you constipated. You don't poop as much, and you end up acidic. Which weakens your kidneys more and makes your brain foggy. Ketosis, oh, you lose weight because you're in a disease state like diabetes. Long-term studies show it kills you. All right, so healthy blood, you want to eat your fruits and vegetables. Unhealthy blood, stay on the animal products. Oh, and your microbiome changes, which determines your emotions, your emotional state. Those little buggers, half of your cells. The advanced glycation end products, class 1 carcinogen. Bacon, smoking, and plutonium. Means it's guaranteed you get cancer. Everything else is a probable or possible or not classified or not. Probably not or not at all. All right, raw broccoli, not going to kill you. This was another, I have not read his book. I've watched his TED Talk. He's an awesome speaker. He actually turned 40. He was a doctor, or 50 or something, and he went and got his, he was healthy, and he couldn't believe he was like a walking time bomb to have a heart attack. And he started researching it. He's like, what? I want to be around to see my kids graduate. And he could not believe what he found, which was just basically all the cancers, heart disease, everything was linked to how much protein you eat. And that is just a picture that Mama Earth can't survive on the diet that Americans eat. So, we get to it. your 30-day challenge for extra credit, I recommend going with the no animal products. Or if you want to, if you're like a big dairy person, no dairy, including cheese for 30 days. Or the other choice is go with the beans, eat legumes. And often if you add something, that will help you get rid of something. So legumes every day, legumes, beans, lentils. It was actually called the lentil effect. It is Joey's birthday today, so you can send Joey happy birthday wishes, but your microbiome loves this, and it then produces the magic bullet, the short chain fatty acids. You can't take it in a pill. It has to be made in your body, and it can only be made from eating fruit and legumes. So 3 pieces of fruit a day, that's what the birthday boy does every day. You can do a salad every day, eat your greens or put it in your smoothie. You can do a rainbow every day. Go for it. It's a tough one. And that says no fast food for 30 days. Some of you tried that. So I got inspired because some of my students, my other class, they've started theirs. They had to. And they had to do it for 7 days, and they're all inspired to now do it for 30 days. Maybe I'll make you have to do it as part of the Lab 7-day challenge. And then if you want to increase it to 30 days, all right. So it's up to you. There'll be a place in 30 days. All right. So happy birthday, Joey, and blessings to all of you. Sorry, I have to figure out how to end.